So now we'll see in detail about the machinery or how the replication is carried out by using different enzymes. Okay. So the replication, it starts at a point that is called origin of replication. It will not start at a random place, but it starts at a particular point that is called the origin of replication. A unit of replication with one origin. So when there is only one origin, it is called the replicon, unit of replication with one origin. But in eukaryotes, there are many origins, right? So unit of replication with one origin is called replicon. So during replication, what is happening is, so this is the parental duplex, right? So it divides, actually this is called replication fork. The two strands unwind. So first step is unwinding, and they separate by breaking the hydrogen bonds. So here they are linked by hydrogen bonds, right? So these lines, it represents the hydrogen bonds. So first step is the breaking of hydrogen bonds at the point of replication, right? So this breaking of hydrogen bonds is done by the enzyme Helicase. So there are many enzymes which are involved in DNA replication. So the first one is Helicase, right? So the unwinding of the DNA molecule at the point. So here is where the unwinding happens. So the unwinding of DNA molecule at a point forms a Y-shaped structure called replication fork. So it forms a structure called replication fork. So this is a fork-like structure, that is, it divides into two. So first enzyme is Helicase. Then the separated strands, they act as a template for the synthesis of new strands. So this is the point of separation. Now these two, they act as a template. So from this, there will be synthesis of one strand, and from the other strand, there will be synthesis of new strand. Okay. So, and also another important factor is the replication will always happen in the 5' to 3' direction. Okay. It will always happen in the 5' to 3' direction. Then deoxyribonucleoside triphosphates. That is, we know that this DATP, DGTP, and DCTP, and DTTP, that is deoxyadenosine triphosphate, deoxyguanosine triphosphate, and deoxycytidine triphosphate, and thymidine triphosphate. So these are the different nucleotides that are present. They act as a substrate. So these colored structures, you know, A, it's the adenine, and this yellow, it represents the thymine, and the green one is cytosine, and blue one is guanine. So these are all the nucleotides that are present, right? So here these deoxyribonucleoside triphosphate, they act as a substrate. So this is the substrate. Okay, and also it provides energy. So as a result of the separation, so here actually originally there was a hydrogen bonding, right, between the bases. So here these deoxyriboside triphosphate, they act as a substrate, and also they provide energy for polymerization, because the polymerization reaction needs energy. So where the polymerization is happening, the new strand is formed, and here they should form a polymer. So it needs an energy, and this energy is provided by the deoxyribonucleoside triphosphates, and also they act as a substrate. Okay. And for the polymerization reaction to start, they need the molecule that is called a primer, that is RNA molecule that is called a primer. A small RNA primer is synthesized in the presence of an enzyme primase. So it is type of a RNA polymerase, that is, it is synthesized by the primase. RNA primers are formed. So first, a small RNA primer is synthesized in the presence of an enzyme primase. So if you see this, can you see from? So this green color area. So this is where the DNA synthesis starts. So now separation and the DNA synthesis starts. So this green is a RNA primer, and here they form a nucleotide, and the polymerization occurs. So this is the RNA primer which is synthesized by primase. Okay. So actually after the unwinding, that is by the breaking of hydrogen bonds by the helicase, there are some proteins which are called single-stranded binding proteins. Single-stranded binding proteins. These which is commonly called SSB, single-stranded binding proteins. And these proteins, actually, it prevents these proteins will come and bind here in the helix, that is the DNA helix, which was unbound. That is after the breaking of hydrogen bond, there is a possibility of rejoining. Right? Again, the hydrogen bonds may form. So these single-stranded binding proteins, it will bind to this single-stranded DNA that is formed. So it prevents the formation or the reformation of the hydrogen bonds. So this is the role of single-stranded binding proteins. And then next, there is another enzyme which is called Topo Isomerase, which is also called DNA Gyrase in prokaryotes. Is also called DNA Gyrase, and main role of this enzyme is it releases the tension. Release the tension arises due to supercoiling. So further, as a result of this unwinding process, there will be more coiling here, and that is relieved by means of the Topo Isomerase enzyme or the DNA Gyrase enzyme. So these are all the different enzymes and proteins which are involved in DNA replication process. Okay. And then the main enzyme that is involved is DNA polymerase. So first step is there should be a separation of the DNA strands and the breaking of hydrogen bond that is done by helicase. After the action of helicase, the single-stranded binding proteins will bind to the single-stranded DNA, and it prevents the reformation of the hydrogen bonds. And then Topo Isomerase, actually, it helps to release the tension that arises. So as a result of this unwinding process, there will be a tension created here. So for example, if there are two threads tied together, so there is a break in one place, there will be more coiling in the other place. So that is relieved by the Topo Isomerase enzyme. And then the new synthesis, that is the synthesis of the new strand starts, that is by the enzyme primase. So first they need an RNA primer for the synthesis of DNA. So the next step is the DNA polymerase. Okay. So in the presence of an enzyme, DNA-dependent DNA polymerase. So here these nucleotides, they join with one another to form, that is the nucleotides are present now, right? So in the presence of DNA polymerase, many nucleotides, they join with one another to the primer and they form a polynucleotide chain or the new strand. So how that is happening? So first step is the primer. From the primer, now new nucleotides are added by the enzyme DNA polymerase. Okay. So during replication, one strand is formed as a continuous stretch in the 5' to 3' direction. So always replication occurs in the 5' to 3' direction. That is called the continuous synthesis, and this strand is called the leading strand. So which is the leading strand there? So if you observe the diagram, this is synthesized continuously. So this is called the leading strand from the 5' to 3' direction. Okay. And the next strand, this is synthesized in a form of fragments. So these fragments are called Okazaki fragments, and actually these Okazaki fragments, they are also synthesized in the 5' to 3' direction, from 5' to 3'. So replication always occurs in the 5' to 3' direction. So other strand that is formed in small stretches called Okazaki fragments in 5' to 3' direction, that is called the discontinuous synthesis. That is, it is not synthesized continuously. Okay, so that is a continuous synthesis and this is a discontinuous synthesis. Then the Okazaki fragments are then joined together to form a new strand by the enzyme ligase. So these fragments are now connected by the enzyme ligase because they are synthesized in fragments and they are joined to form a new strand by DNA ligase. So DNA ligase is a joining enzyme. Okay, so this new strand is called the lagging strand. So that is a leading strand which is continuously synthesized, and the strand which is synthesized in fragments, that is called the lagging strand. Okay, so now if during the synthesis any wrong base is introduced, for example, so this is the process, so this is the original template DNA, so this is the point of replication fork, and these are the nucleotides. And now after the primer is synthesized, so this DNA polymerase, it helps in the addition of nucleotides and helps in the synthesis of new strand, right? So this is the leading strand and this is the lagging strand which is synthesized in fragments, and then these fragments are connected by DNA ligase. Okay, so you're clear with the process, right? So here, if a wrong base is introduced in the new strand, the DNA polymerase can do proofreading. That is, it can also correct. That is, because if a wrong base is introduced, what will happen? That will lead to mutation, right? So the DNA polymerase itself has the proofreading activity that it aids in DNA repair. It will try to correct the mismatched base pair. Okay, actually in the case of prokaryotes, there are three types of DNA polymerase. One is DNA polymerase 1, 2, and 3, which are mainly involved in DNA replication. But this DNA polymerase 3 is mainly involved in that is, it is the main replicase enzyme that is involved in the addition of nucleotides. And DNA polymerase 1, actually this is involved in repair. Actually, it was discovered by Kornberg and it is called Kornberg enzyme, and it is involved in the repair, and also it has an exonuclease activity. As an exonuclease activities. So this is involved in repair, and the main role of DNA polymerase 2 is also, and this is less reactive, and this is also involved in repair in the absence of DNA polymerase 1 and 3. So 3 is the main enzyme which is involved in the replication process.